Thank you to Advance CTR and the organizers for inviting me to present this work today. I'm Kathryn Grive. I'm an assistant professor of obstetrics and gynecology. And I'm going to first start with an overview of the work going on in my lab and to orient you to our interests. And the topic I'll be presenting today is effects of maternal cancer therapy on daughters' ovarian health and fertility preservation. So we have a lot of projects going on in the lab, and I can't cover all of them today, but historically, research on my lab has focused on ovarian physiology and oncofertility. And so we're interested in understanding mechanisms of chemotherapy-induced oocyte damage. These are what is colloquially known as eggs in the ovary, so I'll use eggs and oocytes interchangeably. We're also interested in elucidating the long-term reproductive effects of in utero exposure to cancer therapeutics and to identify novel regulators of oocyte health and survival, which we may be able to leverage this knowledge for the preservation of the ovarian reserve and via fertility preservation efforts. Today I'm going to be telling you specifically about the effects of in utero exposure to cancer therapeutics, which was funded by my Advance CTR Pilot Award. First, I want to start with a brief introduction to the importance of the mammalian ovary. Put simply, the mammalian ovary is composed of both oocytes, again eggs, we sometimes also call these germ cells, as well as ovarian somatic cells. And its primary functions are to produce the oocytes, which are the female gametes, as well as steroid hormones such as estrogen and progesterone. Folliculogenesis is a specific process that takes place in the ovary in which oocytes and somatic cells develop in concert with one another, and that schematic is shown at the bottom, to produce oocyte-containing follicles. And as you can see in this schematic, the follicles will develop through the primordial, primary, ultimately antral stages, and during this time they acquire increasing organization of the surrounding somatic cells. Ultimately, there will be a dominant follicle produced in the middle of the cycle, and it can be ovulated for fertilization. When this process is dysfunctional, it can lead to reproductive pathologies, specifically a condition known as primary ovarian insufficiency or premature ovarian failure is a dated term for that condition, as well as other reproductive dysfunction. And this is so critical because not only does oocyte abundance affect a woman's ability to conceive, but it impacts the endocrine function of the ovary, which directly affects women's overall health, including their actual lifespan and other features of aging. So all ovarian function is predicated upon the successful establishment of the finite and non-renewable population of oocytes that we know as the ovarian reserve. This is established in utero in most mammals. So I'm going to be showing you a timeline of this development both in mouse and in human. The mouse timeline is on the top and the human timeline is on the bottom. In the mouse, germ cells, those developing oocytes, arrive at the developing gonad around embryonic day, or E10.5. So this is right in the middle of mouse gestation, which is about 20 to 21 days. There they will undergo mitotic divisions with incomplete cytokinesis, forming cysts. And after this mitotic program, meiosis commences around embryonic day 13.5. The oocytes will progress through the beginning of meiosis and ultimately become arrested in meiosis around the time of birth. Around this time, cysts also undergo a process known as cyst breakdown, and the fragmenting of these cysts results in a massive wave of oocyte loss. The remaining oocytes will then become surrounded by a single layer of somatic pregranulosa cells, forming the ovarian reserve. And this ovarian reserve then has to remain quiescent and stable for a very long period of time, up to decades in humans, prior to being activated and ovulated. And you can see on the bottom, the human timeline, this happens very early in gestation as well. So what we do know about the process of ovarian reserve establishment is that it can be affected in a variety of ways. And one of those ways is through exposure to environmental insults. And so for today, I'll be focusing on this in the context of cancer therapies. But what I want you to take away from this slide is that while the ovarian reserve represents the entire reproductive capacity of a female, We know very little about the regulation of its health or targetable pathways to prevent its loss. We know a lot about cancer therapeutics, specifically chemotherapies, and how these can affect the ovarian reserve when they're administered to adults, but we know very little about how these can influence prenatal development. And this is of increasing concern, as currently approximately 1 in 1,000 women are diagnosed with a malignancy during pregnancy. The rates of maternal malignancy are rising with an increasing median age for childbearing. The majority of these cancers are cervical cancer and breast cancer, but other cancers such as melanoma, lymphomas, and leukemias are relatively common as well. A study published last year out of the United Kingdom presented the latest demographics and current treatments for patients diagnosed with cancer during pregnancy. And these data showed that the average age was the early to mid-30s. These are mostly diagnosed in stage 1 and as a new cancer. The majority of these are in the 2nd or 3rd trimester, and the greatest proportion of them are breast and skin cancers. So how are these cancers treated? If a provider determines that a woman should seek treatment in the 1st trimester, and this is, of course, a case-by-case basis weighing the risks and benefits, then the patient's usually counseled to terminate the pregnancy, as the processes associated with organogenesis can be so gravely affected by many of these agents. But in the 2nd or 3rd trimesters, treatments are often administered to pregnant women at the provider's discretion, if they determine that the benefits do in fact outweigh the risks. Importantly, chemotherapies are known to cross the placenta to varying degrees depending upon their size. And again, in the postnatal context, chemotherapy is known to lead to excessive oocyte loss and infertility, including, as I mentioned before, the development of primary ovarian insufficiency, which is that premature loss of ovarian follicles, which can then result in not just infertility, but loss of the ovarian steroid hormone production and ovarian function itself. And again, overall women's health relies on the proper functioning of the ovaries to support endocrine balance. So open questions in the field, therefore, are: do chemotherapeutics exhibit in utero consequences on the developing fetal ovary? If so, what are long-term effects of chemotherapeutics on the ovarian reserve? What is the effect on the fertility potential of a child and grandchild of someone who underwent chemotherapy while pregnant? And which chemotherapeutics are least harmful to the ovarian reserve of the developing fetus? So our overall goals are: we aimed to identify the effects of in utero exposure to chemotherapy and immuno or targeted therapies on the fertility potential of exposed daughters. To go back to the UK maternal malignancy study, we see that among the most common treatments are taxane-based chemotherapies. These alter microtubule polymerization or stabilize microtubules and ultimately inhibit cell division. Docetaxel and paclitaxel are among these agents. They're commonly used in breast, lung, and ovarian cancers. There is no evidence to suggest compromise of fetal health based on the studies that exist. And we do see that there's about a 2 to 9% placental transfer. Because they cross the placenta at lower rates than other therapies, we hypothesize that they may be more fertility sparing for daughters who are exposed in utero. We also see the use of monoclonal antibodies or immunotherapies. These target unique features of cancer cells or stimulate the immune system to recognize, attack, and eliminate tumor cells. So of interest to us was anti-cytotoxic T lymphocyte-associated antigen, CTLA-4 therapy, which is commonly used in melanoma, lung, colorectal, and other cancers, as well as anti-vascular endothelial growth factor or VEGF therapy, which is commonly used in breast, lung, colorectal, and other cancers as well. So our experimental design utilized a mouse model of pregnancy to begin to explore these questions. So to do this, we use time matings, which allow us to accurately determine the gestational age of our pups from a given cross. We then administer a single animal equivalent dose. So this is a therapeutically relevant dose for human treatment at embryonic day 16.5. or what's the equivalent of the 2nd to 3rd trimester ovarian development for a human. And then we allow these pups to be born, and we collected daughters' ovaries at postnatal day 14, which is prior to sexual maturity. So these are pre-pubertal animals, or postnatal day 30, which is after sexual maturity. And in these samples, we analyzed ovarian follicle composition, or as well as follicle atresia. So this is sort of follicle death. We also set aside additional exposed daughters as breeders to determine the effect of these exposures on fecundity. And since the beginning of this study, we've had a chemotherapy cohort, as well as the respective controls, as well as a monoclonal antibody cohort and respective controls, but I'll only be telling you about the chemo groups today. When we compare taxane-exposed ovaries to controls, we see a decrease in ovarian area initially, but this seems to have recovered by the time of sexual maturity. So here again, we're comparing that pre-sexual maturity or pre-pubertal time point at postnatal day 14 to the post-sexual maturity or sexually mature animals at postnatal day 30. And at the time of sexual maturity, we don't see any obvious effects on oocyte density as well. When we analyze follicle density in these animals, while we don't see any changes in the prepubertal group, we do see a non-significant decrease in the primordial follicles of the ovarian reserve in taxane-exposed sexually mature animals, along with a commensurate significant increase in growing follicles, suggesting that there may be an accelerated or inappropriate follicle activation in these animals. It's also possible that taxane exposure may inhibit the expansion of the follicle cells during follicle growth, meaning that these follicles are sort of piling up at this immature stage, unable to progress further through maturation, or at least not at a normal pace. When we look at the actual histology of these ovaries, you can see healthy ovaries, which have been exposed to saline in utero in this first panel. But what I hope you can appreciate is that in both the docetaxel and paclitaxel exposed groups, we see altered follicle architecture as well as degenerating follicles, and that's what you're seeing, sort of those structures in the interior of the ovary that have space around them. Those are follicles that contain an oocyte but are sort of collapsing in on themselves due to that process that I referred to earlier called follicle atresia. And we see this at a much higher rate in the chemotherapy exposed groups compared to the saline groups. When we quantify this, we see a significant increase in follicle atresia in both chemo groups at both time points, and this coincides with increased tunnel positivity, which demonstrates apoptotic cells, and this is in the follicular cells of the follicles of these groups, indicating, again, the presence of apoptotic granulosa cells, or those somatic cells, and is indicative of unhealthy follicles. Furthermore, we see an increased incidence of multi-oocyte follicles in the docetaxel exposed group specifically. This is a phenotype in which two oocytes are contained within a single follicle, so a single basement membrane. As a result, neither oocyte will receive sufficient somatic support for developmental competence and will not be viable as a result. So next we wanted to ask about the function of the hypothalamic pituitary gonadal axis, the HPG axis. And this is a feedback loop in which steroid hormones such as estrogen and progesterone, which are produced from ovarian follicles, feed back to the hypothalamus in the brain and then to the pituitary gland. Those in turn will release gonadotropins such as follicle stimulating hormone and luteinizing hormone, and these will participate in the feedback loop that regulates the menstrual cycle in humans or the estrous cycle in mice. And any disruption to any of these inputs will then disrupt that proper cycling. So anti-Müllerian hormone is produced by the granulosa cells of ovarian follicles, and it's a surrogate marker for follicle abundance. So consistent with our finding that there's no change to oocyte density across groups, we see that there is no change in AMH levels across groups either. In contrast, we see significantly different FSH levels between treated groups compared to our controls. Interestingly, they seem to behave somewhat differently, suggesting that both disrupt the functioning of the HPG axis, but perhaps in different ways. So high FSH in the docetaxel group is consistent with premature ovarian aging or a menopause-like phenotype, whereas low FSH in the paclitaxel group may be indicative of altered levels of other hormones that are not profiled here. Specifically, that may be gonadotropin-releasing hormone from the hypothalamus, which is a known target of taxane toxicity. Additionally, we stimulated animals with gonadotropins to induce ovulation, much like what is performed on humans prior to an egg retrieval for IVF. And that's to test the ability of these exposed animals to ovulate properly. And we see that both treated groups ovulate significantly fewer oocytes in response to this hyperstimulation, which is again consistent with HPG axis disruption. Finally, when we perform breeding studies with exposed animals, we see that both taxane exposed groups produce fewer litters over the course of the study compared to controls. But when they do produce litters, they are of normal size and have normal viability, suggesting that the exposure has affected their ability to ovulate normally, but does not affect their ability to carry healthy pregnancies. Recently, we've begun to look at the transgenerational effects of these therapies. Remember that the ovarian reserve is established in utero, so while a fetus is being exposed to therapy during gestation, her oocytes, which will ultimately comprise the next generation, are also being exposed. Therefore, it's conceivable that the granddaughter of a pregnant woman being treated for cancer could experience long-term effects of this exposure. So when we analyzed ovaries of the F2 generation, so these are the daughters of the mice exposed in utero, or the granddaughters of the mouse that received chemotherapy, we found that while folliculogenesis is in these prepubertal mice was unaffected, we did observe a significant increase in degenerating growing follicles in the docetaxel group, but this was not seen in the paclitaxel group. And again, here, I hope you can appreciate these histology images showing that there is indeed this very obvious follicle atresia in the docetaxel group. Again, these are the granddaughters of the mouse that was originally exposed to chemotherapy, which was a really kind of exciting and somewhat surprising effect that we observed. So in conclusion, daughters exposed to taxane therapy in utero experience altered folliculogenesis, increased follicle atresia, decreased fecundity, as well as endocrine dysfunction. These effects on follicle atresia were also observed in the F2 generation without the direct exposure of these animals to that agent. It was just their developing oocytes. And that's perhaps due to epigenetic changes as well as potentially other mechanisms. Taxane exposure can affect the ovary in distinct ways, with docetaxel generally producing more deleterious effects. We do know that docetaxel and paclitaxel have slightly different mechanisms of action. Docetaxel exhibits a greater ability to bind to beta-tubulin and inhibit microtubule depolymerization, whereas paclitaxel specifically affects organization of the mitotic spindle. Given that oocytes are no longer mitotic, it's possible that that may be a reason that paclitaxel is slightly less deleterious to that population. And then I mentioned that monoclonal antibody cohort, which I didn't have time to present to you today, but breeding studies and ovarian analysis are currently underway for the offspring exposed to anti-CTLA4 and anti-VEGF targeted monoclonal antibodies. And we're doing basically this exact same series of experiments and analysis to understand how these agents may affect long-term reproductive function in daughters and granddaughters exposed in utero. So with that, I want to acknowledge my lab, who have done, obviously this work. The names bolded here are the people who have done the lion's share of the work. I also want to acknowledge my sister lab in the program for women's oncology at Women and Infants Hospital. This is Dr. Nicole James's lab for molecular therapeutics, and she does a tremendous amount of work on ovarian cancer and is well-versed in molecular therapeutics for cancer, and so we work very closely with them. I also want to acknowledge my funding, which has come from many sources for this project, but I specifically want to thank Advance CTR for their support of this pilot, which has been really exciting to undertake and is really kind of the first set of data, especially on transgenerational effects of in utero exposure to chemotherapeutics that the literature has seen. So I again thank you for having me here to present this work today and for your attention.